Now we're going to discuss transcription. The transcription is a synthesis of RNA based on using one strand of DNA as a template. First step in gene expression. So first we'll discuss RNA structure, the types of RNA, then transcription in prokaryotes, then in eukaryotes, then reverse transcription, and finally RNA replication. So remember that RNA is single-stranded, it's uracil instead of thymine. The other three bases are the same. Excuse me. Here's a table comparing DNA and RNA, and you can read through that just as well as I can, right from the textbook. Here, remember RNA is a single-stranded molecule. 3 of the 4 bases are the same as DNA. Here's a uracil nucleotide, and notice the 2' OH there that is lacking in DNA. So the primary structure of RNA would be the nucleotide or base pair sequence, and the secondary structure would be then how the whole molecule folds around on itself. So there are 3 RNAs that are present in both prokaryotes and eukaryotes that are all important in the process of translation or protein synthesis. Messenger RNA, ribosomal RNA, and transfer RNA. And you can read what they do as well as I can. This shows you that all cells have those types, and then there are several other more specialized RNAs in eukaryotes only. We won't talk about those too much, but we'll allude to them once in a while. And this again is a table from the book talking about what some of these different RNAs do. We're going to focus on the 3 top ones: ribosomal, messenger, and transfer RNAs. So, just like DNA replication, transcription has 3 steps as well: initiation, elongation, just adding more nucleotides, and then termination of the process. So in prokaryotes, transcription and translation can occur simultaneously, but they cannot in eukaryotes because in eukaryotes the DNA is walled off in the cell nucleus. So transcription occurs in the cell nucleus, and then the messenger RNA is transported to the cytoplasm where translation can occur, as we see in the lower diagram there. So in prokaryotes then, transcription begins with the RNA polymerase, which is the molecule responsible for synthesizing RNA, binds to promoter regions. The RNA polymerase covers about 60 base pairs of DNA. The promoter regions are these short DNA sequences just upstream from the gene. As you can see here, here's the core RNA polymerase, and it needs the sigma factor, another protein entity, to bind to it for the enzyme to be completely functional. This shows the DNA as a ball model and the RNA polymerase protein as a ribbon model attached to the DNA. And notice how the DNA bends there where it attaches. So this shows you in yellow is the promoter region, and then the RNA coding region, or the gene or genes, is shown there in the purple color. Here are a couple of promoters, one at minus 35 and one at minus 10 base pairs before the gene that's going to be transcribed. This defines what a consensus sequence is. It's the most commonly encountered nucleotides at each site. So the consensus sequence there at the bottom, where T and A are actual nucleotides, Y means a pyrimidine, and R means a purine, and an N means that there's no particular base in common, and that CG notation means that C and G are equally common at that site. And so here are promoter regions from various prokaryotic genes showing you that they are similar in their sequence but not identical, with the consensus sequences at the bottom. So our core RNA polymerase then binds to the promoter region, and then the DNA begins to be unwound there, forming a transcription bubble, and it proceeds in a 5' to 3' direction, just like DNA replication. But prokaryotes, unlike us eukaryotes, can transcribe several genes at a time. Eukaryotes transcribe only one gene at a time. This shows you the transcription bubble with the red RNA transcript coming out of the top there. The green blobbish thing is the RNA polymerase enzyme, and then of course the DNA there, which is locally unwound where replication is transcription is occurring. Excuse me. So elongation, the template strand, the anti-coding or non-coding strand is used to make the RNA. The coding strand, then the other strand has the same sequence as the RNA, except of course it's got the DNA has T's, and whereas the RNA has U's in it. And nucleotides are added in the 5' to 3' direction only, just like the DNA replication. Unlike DNA polymerases, though, RNA polymerases don't have proofreading ability, but as we'll see later, that doesn't really matter that much. So then this shows you that the non-template strand will have the same sequence as the RNA. The template strand will have a sequence complementary to the RNA. And this shows you the process continuing. And of course, base pairing between DNA and RNA is very similar to between two strands of DNA. This shows again the the RNA strand in red there is complementary to the template strand and has essentially the same sequence as the non-template strand, except that it has U's instead of T's. And so here we can see the ribonucleotide triphosphates that exist in the cell. When they're not incorporated into the RNA molecule, they exist as triphosphates. And this shows several RNA molecules being made from a single DNA molecule. So you can see there, look like little Christmas trees. Now, termination. There are two types of transcription terminators in prokaryotes, and they both are have inverted repeats separated by about 4 base pairs. They're palindromic. Palindromes are things spelled the same way backwards or forwards. Hairpins often form to slow the RNA polymerase. One is referred to as a row-independent terminator. The other is a row-dependent terminator, where row is a type of protein involved in the termination process in row-dependent termination. And we'll discuss how each work. So row-independent, the inverted repeats form a hairpin. This hairpin causes the RNA polymerase to pause. The UA base pairs right after the hairpin, only 2 hydrogen bonds. And so these hydrogen bonds break, and that frees the RNA transcript, the DNA, and the RNA polymerase molecules. And it looks something like this. So here's our inverted repeats at the top. And we see at the bottom the RNA transcript still attached to the DNA there. And then it forms the hairpin. And that causes the RNA polymerase to stall. And so that releases the RNA transcript, and the DNA winds back up again. And of course, the RNA polymerase is released as well. And here both those slides in one. Now, the row-dependent terminator needs the row protein. It lacks the TAs after the hairpin. The row attaches to the 5' end of the RNA molecule being made and moves up. And this the RNA polymerase pauses after the hairpin. At that point, the row protein unwinds the DNA, the RNA, and the RNA polymerase, and transcription is terminated. If there is no row protein, then the terminator sequence is ignored, and what we have there is called read-through. So transcription just keeps continuing. So this shows you how that works. There's the row protein moving up from the 5' to the 3' direction up the RNA transcript, and then when it gets to the RNA polymerase molecule and the transcription apparatus, it unwinds everything. This shows what's called a polysome that's present in prokaryotes. You have DNA running through the middle there, as you can see labeled over towards the left, and then there's messenger RNA molecules snaking off that DNA, and the little ball-like things attached to them are the ribosomes that are already starting translation. So here we have transcription, translation occurring simultaneously, and that's called a polysome, and it only happens in prokaryotes, not in us eukaryotes, because of course our DNA is inside the nucleus. Translation occurs out in the cytoplasm. Now our eukaryotic transcription is monocistronic. Cistron is another term for a gene. So in other words, we transcribe one gene at a time, not clusters or groups of genes like prokaryotes do. So this shows that transcription occurs in the cell nucleus on the left there, and then the finished RNAs then are shipped out of the nucleus into the cytoplasm to take place in translation, which is on the right side of this diagram. Now, eukaryotes have several RNA polymerases. RNA polymerase 1 transcribes ribosomal RNA, including the 3 large ribosomal RNA genes that make up the NOR or nucleolar organizer region. RNA polymerase 2 does the messenger RNA genes. RNA polymerase 3 does the small ribosomal RNA genes and transfer RNAs. And there's at least a 4th and possibly a 5th one, but we're not going to talk about those. So anyway, there could be as many as 5 different RNA polymerases in us eukaryotes. This shows an RNA polymerase where the RNA polymerase 3, where the nucleotides are funneled in through the bottom, and the RNA transcript and the DNA sort of snake out the top. The DNA is fed into the right side, as we can see there in that groove. So eukaryotic promoter regions include the Hogness or Tata box at -25 base pairs before the gene, the cat box at -70 with a consensus sequence that has CAAT in it. RNA polymerase 2 binds to the cat box and moves down to the Tata box. Transcription factors are numerous proteins that help, and they tend to bind to promoter regions and other regions also. They're like enhancers, which can be several hundred, even several thousand base pairs away from the gene. And special transcription factors bind to the enhancers, where basal transcription factors bind to the core promoters, the things at the top of this slide here, like the Hogness and the Tata box. And so here are the core promoter regions, just upstream of the gene in eukaryotes. Here are different core promoter regions for 3 different eukaryotic genes. So here's initiation. So the long hot dog-like object is the RNA polymerase 2, and then all those other things that say TF are transcription factors. So there's many, many of them. And then you can see at the bottom there an enhancer that causes the DNA to bend around. So special transcription factors bind to the enhancer, basal transcription factors bind to the RNA polymerase and associated core promoter regions. Now terminators for eukaryotes. So the different RNA polymerases use different terminators. RNA polymerase 1, there's a termination factor that binds the DNA downstream of the termination site. On RNA polymerase 3, they have termination like row-independent terminators in prokaryotes, but without the hairpin. Then RNA polymerase 2 often synthesizes the DNA from hundreds of thousands of nucleotides past the coding sequence. And the 3' end of the mRNA is cut at a consensus sequence. And we've got 2 pieces of messenger RNA that result from this process. The pre-messenger RNA transcript, and then the RNA with the 5' end coming out of the RNA polymerase 2 molecule. So a protein called RAT1 It's an enzyme that attaches to the 5' end of the RNA that's snaking out of the RNA polymerase II molecule, moves up towards the 3' end, and it has exonuclease activity, so it can degrade or chop up the RNA as it moves up. Once it reaches the transcription complex, transcription is terminated. And this shows that process with in number 1 there, RNA polymerase II transcribed well past the coding portion of the gene. Cleavage then is near a consensus sequence while the RNA polymerase pauses, or while the RNA polymerase continues transcribing. Then RAT1 attaches to the RNA that's coming out. The messenger RNA transcript has already been cut off at this point. And the RAT1 endonuclease attaches to the 5' end and moves up, degrading that remaining RNA. And then when it gets to the transcription apparatus, the whole thing is unwound. And so this shows the whole process that's shown in the last 2 slides on the same slide. So that's transcription in eukaryotes. Now we're moving on to reverse transcription. So on the right here, we see that on retroviruses, they can use RNA as a template to make DNA. And we're also going to talk about RNA replication as well. So here's a retrovirus showing you the reverse transcriptase molecules that are brought into the cell along with the single-stranded RNA genome. This is the RNA genome, the genome of a retrovirus, I believe an HIV virus, showing you there's really basically 3 genes, as we can see in the bottom there. GAG that codes for a viral capsid protein, POL that codes for integrase and reverse transcriptase, and then ENV that codes for envelope proteins. So the longer name for reverse transcriptase is DNA-dependent RNA polymerase, but it's just always called reverse transcriptase. Viral RNA brings this enzyme into the cell. The enzyme makes a DNA-RNA double helix, which then gets converted to a DNA-DNA double helix, which then gets integrated into the host's genome. And so now we've got the virus's genes in our genome. And of course, when they get turned on, it makes viral proteins. So this shows you that process where the virus fuses with the cell membrane, and then the process of transduction where its genome comes into the cell along with the reverse transcriptase enzyme. And we see the reverse transcription process occurring in the cell cytoplasm. And then finally, the double-stranded DNA gets moved into the nucleus where it's incorporated or integrates into our genome. And then when it's activated, it produces viral RNA, which undergoes translation to make the other proteins to assemble new virions. And as they bud off the cell, of course, that lyses or kills the cell. This shows you the integration process where the formation of recessed 3' termini is cleaved by enzymes called endonucleases that cut the sugar phosphate backbone. And then that allows the viral DNA to attach there, and host enzymes repair the little attachment areas. And voila, we've got new genes in our genome. However, they're genes we don't really want. Now, with regards to RNA replication, this is RNA copying itself. It occurs in some simple viruses and some transposable elements. For instance, a virus called MS2 has 3,500 base pairs and 3 genes. 1 gene codes for coat protein, a 2nd gene codes for attachment protein, and a 3rd gene codes for a single subunit of RNA replicase. And what happens is that single subunit of RNA replicase combines with 3 cell proteins to form functional RNA replicase that then replicates the virus's RNA in the host's cell. And that is all we have concerning transcription.